Alan Tabler. My name is Alan Tabler, and I'm here as the host representing the North American branch of the Maple Society. Dr. Jake Grossman is a visiting assistant professor at Swarthmore College, where he teaches courses on ecology, plant biology, and global change biology. He holds a PhD in ecology, evolution, and behavior from the University of Minnesota. His love of maples was nurtured by a 2-year postdoctorate fellowship studying the tree physiology at the Arnold Arboretum, where he worked with the Arboretum's renowned Acer collection. Ladies and gentlemen, Mr. Jake Grossman. Take it away, Jake.
All right.
Thank you so much, Alan, and thank you all for taking some time. I realize for some of you it's a little bit late, as it is for me. I am really excited to get to principally share some of the research that I've done on the global maples with you all, given our shared interest. Just a few housekeeping details before I share my screen and start. I'm going to mute the people who are not muted before I start my presentation, just to minimize background noise. If you have a pressing question during the presentation, you're welcome to unmute yourself and ask. I think my preference would be to save questions, if possible, for the end of the presentation, so we can have a more organic conversation. But if there is something that you really would like to ask or say, feel free to do so. If you are having technical issues, you can also chat about them, and I'll try to keep my eye on it. And Alan is also a co-host and can maybe help me out if something manageable comes up. So if there are no concerns, I'm going to just mute the folks who are not yet muted, and I will get started. All right. All right, so can folks see my PowerPoint slides? It looks like yes. Okay, so I'll get started. So again, thank you, Alan, for that introduction. I won't share more about myself, except to say that I'm excited to get to chat with you all a little bit about Acer, about maples. So a roadmap for this talk is, I'm going to give a very, very, very brief introduction to the genus. I feel a little uncomfortable doing so because I imagine many of you are more knowledgeable about its natural history and diversity than I am. But I'm keeping this segment in my talk in part to share some terminology that I'll be using and to update us all on what I consider to be the state of the science surrounding maple evolution, or our understanding of maple evolution. And then the bulk of my talk will be covering what is, I hope to many of you, new information, which is the research that I've been doing on the maple genus over the last 3 or so years. All right, so I'll start with this brief orientation to the genus's natural history. So many of you are probably familiar with this illustration showing us that the maples are truly globally distributed genus across the temperate north. And I like to think about them, though there are some exceptions to this pattern, as belonging to either the floras of East Asia and Southeast Asia, the East Asia being the center of diversity of the maple genus, Eurasia, or North America. And I'll refer to these biogeographic labels, simplistic as though they are, later in the talk. Depending on how we splice them, I guess I tend to be conservative and lump species together. Others prefer to split species and have a more liberal view. There are about 120 to 160 species in this genus as currently described. And as many of you may know, the maples were recently reclassified out of their own family into the Sapindaceae, the soapberry family. This this phylogenetic tree or evolutionary tree is an old one. It's not the most recent, but I use it because it shows a wide variety of maple species, as well as, and this is what I will focus on here, their relatives outside of the maple genus. So as is still the case, it is was believed when this paper was written that Acer was sister or more closely related to the genus Dipteronia than to any other genera, and that these clades are themselves closely related to Aesculus, the Buckeye horse chestnut group that many of us are probably familiar with. So these are some of the close relatives of the maples as far as we understand now. I do want to spend some time on this new account of the evolution of the maples, and I'm going to take a moment to explain how to interpret this figure for those who are not used to reading, again, what are called phylogenetic trees, and that's just a fancy way of saying an evolutionary tree. So I'll use my cursor to explain this. In this tree, which we can read from right to left, each line on the right-hand side is what's called a tip. And each of these tips represents an existing and a non-extinct maple species. These are not all 120 to 160 species, but rather a sampling from across the genus that tries to capture its diversity. And all of the tips end at the same place because this evolutionary tree has been calibrated against fossils of maples such that as we move from right to left across the tree, we go back in time. And this is labeled at the bottom of the tree. It's a little hard to see for these briefer epochs, but we have the Pleistocene and Pliocene, and then Miocene, Oligocene, Eocene epochs, etc. So we're going back about 75 million years to around the time when the maples split off from the outgroup or their nearest ancestors, the plants that were to become the Dipteronias, the Aesculus, etc., shown here at the bottom of the screen. What we can then infer from this illustration is that wherever there is a node or a circle, two species or two groups of species of maples diverged. They went their separate ways evolutionarily. So clusters of nodes that are very far to the right, like these orange clusters that I'm indicating right now, are relatively recently diverging from each other, which means they evolved to be different in the relatively recent past, maybe only 5 million years ago. Whereas a very deep division or node in the tree, like this one I'm indicating, occurring over 50 million years ago, represents a very ancient division, one that we might assign to the difference between two very different sections of maples. This tree was constructed using evidence from the genome of well over half of the known maples or the described maple species. And interestingly, for those of you who do ever think about genetics, evidence for this tree was harvested from two different sources. One was the nuclear genome, which what we normally think of when we talk about our genes, which is the genome that we, or plants for that matter, inherit equally from both of our parents, as well as evidence from the chloroplast genome. This is the genome that encodes the information plants need to make chloroplasts, which they use to do photosynthesis, and it's only inherited from a plant's maternal ancestor, its mother, which makes it more conservative. It doesn't change as rapidly, and it provides a different type of information than the nuclear genome. And so combining these two forms of information, Areces Berazain and colleagues in this very recent paper published this year constructed this evolutionary tree reflecting what we now think of as the history of the maple genus. And in addition to doing all of that, they very helpfully assigned each of the species in this tree to a biogeographic region. Here they distinguish 5. Yellow and orange represent the East Asian groups, green represents Eurasia, and light and dark blue represent North America. And this allows us to begin picking apart some interesting patterns in the evolution of the maples. So in this paper, the folks who put this phylogeny together have drawn on past work and merged it with their own findings to validate the existence of 15 sections, or excuse me, of 17 sections of maples. 15 of these, which I've highlighted in lime green, have simple leaves, which we know is the most common leaf form for maples. An additional 2 sections have compound leaves, suggesting either that compound leavedness, the condition of having compound leaves, has evolved twice, or that it was the original state of the maple common ancestor, and that meant the other 15 sections have converged towards having simple leaves. And the 2 compound leaf sections include this small one at the bottom of this particular tree, the box elder section, which includes box elder from North America, as well as a few Asian species, and then the trifoliate maple section, which is a bit larger. And just to orient you a little bit in this phylogenetic tree, some notable elements we can see here include at the very top of the tree, the Eurasian American sugar maple group. So this includes in light and dark blue, the Acer saccharum and the various species or subspecies associated with that complex, as well as all of its closest relatives, which interestingly are distributed in Eurasia, suggesting that unlike the other North American maples, sugar maple's ancestor arrived over a land bridge going through what is now Greenland rather than through a land bridge going across what is now Alaska. Also of note here is a pair of closely related species that only diverged about 15 million years ago, and that are, I would argue, the sort of truly North American maples. These are the red and silver maple, which are very closely related to each other and much more distantly related to any other maple species. And just to sort of, again, incorporate some natural history into this account of maple evolution, I've highlighted in yellow the Pliocene and Pleistocene epochs, the period of time going back about 5 million years, and that has most directly shaped the climate and the flora that we experience now. This period of relatively rapid cycling between ice ages and interglacial periods that has given rise not to differentiation between what are now different maple sections, but certainly to diversification within groups of what we think of as closely related maples, such as the sugar maple group at the top of this chart. Okay, so I probably don't need to convince anyone else here that the maples are important beyond what you already brought in with you and beyond that account of how fascinating their evolutionary history has been. But I also like to point out that we're justified in our interest in this genus by the critical nature, the critical role that maples play in American and in North American forests. In American forests, for instance, maples are the 3rd most speciose genus behind Quercus and Pinus. And similarly, they comprise the 3rd largest genus in terms of biomass across American forests per USFS data. Okay, so this is really a review, and I'm just going to go through it briefly to make sure that we're all on the same page. But in this talk, I'm going to focus on what I would consider to be the North American maples, making some, relatively arbitrary choices about what counts as a species and what doesn't. So I'll distinguish occasionally between the Eastern North American maples, the ones that I know some of you are most familiar with in our everyday life and in the forests and natural woodlands around us. These include the sister species, red maple and silver maple, some 2 less common understory species, Acer pennsylvanicum and Acer spicata, as well as box elder, which has this very broad distribution across the United States. Further, maple, depending on how we choose to slice the pie up, is either an Eastern North American species with a range roughly coterminous with that of red and silver maple, or a complex that spreads all the way deep into Meso and Central America and to the Rockies. again, I don't mean to be at all controversial in describing the taxonomy and the systematics of this species or species complex. But I'll just note that putative subspecies of sugar maple include 2 southern maples, Floridanum and Leucoderma, black maple subspecies nigrum, which coexists with sugar maple subspecies saccharum throughout its range. Big tooth maple recognized and validated in the evolutionary tree I just showed as a separate species, which is the western sugar maple, Acer grandidentatum. And then 2 Mesoamerican, Central American species, the cloud forest maple, Acer scootchii, and the recently described Mexican maple, Acer binzayetii. And then, of course, we have the, relatively speaking, species depauperate at this point in time, Western North American maple flora, including some isolated populations of box elder, as well as the beloved big leaf, Rocky Mountain, and vine maple. And I know some of you are from or are living in the West Coast, and I want to, I want to make up for the fact that I've sort of given short shrift to the species diversity of your maples by pointing out that we know that in the relatively recent past, the maple flora of Western North America was much more diverse. In fact, in a landmark 1987 study, Wolf and Tenai found evidence of at least 98 species from 28 sections, again, relative to the 17 extant sections across the entire world in Western North America alone. So this is evidence of what was likely a global pattern of a much more diverse maple genus losing diversity as our climate became, relatively speaking, cooler and drier and experienced cyclic ice ages and interglacial periods. Okay, so that's perhaps a somewhat basic review of an introduction to the natural history of the maples. But as a teacher, I want to make sure we're all on the same page before diving into something entirely new. So next, what I'm really excited to get to share with you is what I have been able to contribute to our understanding. And in particular, I'm excited to present this information because it is motivated by my own and what I hope is all of our interest in understanding what the consequences of climate change will be for ecosystems and for plants that we care about. So I like to begin lessons about or discussions of climate change by drawing on this paper published a few years ago in the Proceedings of the National Academy of Sciences that presents what I think is a very sobering and important analysis. So essentially, if we take the worst case scenario or the business as usual scenario for global emissions as estimated now, what we might expect in the next 150 to 200 years is about an 8.5 degree on average surface warming across the globe, shown in this plot. It's really hard to know what that will be like, what this type of surface warming will do, particularly to organisms and ecosystems that we rely on for our well-being and our aesthetic enjoyment today. We know that this amount of warming is likely to cause thermal expansion of the world's oceans, as well as ice cap melting, and will drastically change, of course, climatic conditions across the globe. But what will it do to our ecosystems and populations and communities of living organisms? Well, the authors of this paper used past evidence of paleoclimate and of the organisms that thrived in climates of the past to try to forecast or understand what sorts of ecosystems and communities we might expect under different climate change scenarios. And extremely disturbingly to me, what they found is that if we pursue a business as usual emission scenario, we are on track, again, within 150 years or so maybe 7+ maple tree generations, to create climate conditions that the Earth has not seen for 50 million years, so since the Eocene. And you may remember that this was a time period when the maple genus, like many other common today temperate genera, was just being able to differentiate into the primary sections that we know today. So this is not to say that we're going to experience Eocene flora and fauna in 150 years. Of course, we can't. But it is to say that many of the most important plants and animals to our lives today have evolved since the Earth last, or have undergone some of their most critical adaptations since the Earth experienced this kind of climate. And to me, it provides grounds to be skeptical that some of them can withstand this amount of climate change over a period of 2 centuries, rather than tens of millions of years. So this is what motivates my work. This is a research program that I began as a postdoc at the Arnold Arboretum, and that I've continued at Swarthmore College. I've also broadened it a bit here to take advantage of our magnolia and holly collections, which are, like the Arnold's maple collection, PCN accredited. And I'm happy to chat more about magnolias and hollies later if anyone would like. So here are the questions that have motivated me, and I'm going to share a little snippet from each of them. So the first, and the one that to me is the most pressing, is how vulnerable are the maples to drought? And to answer this question, I'll share data about a particular metric of drought hardiness called Turgor loss point. Second, and maybe a little less intuitively, there's a reason for concern that even though our local climates in many places where maples grow are getting warmer, that may create a paradox in which maples are actually at risk of leafing out early and then freezing when a fairly typical winter storm occurs after a period of warm weather. And this is a phenomenon called false spring. And false springs are thought to be potentially become more common in many climate change scenarios. So I'll present an analysis of maple vulnerability to false springs using a technique called thermal analysis. And finally, I'll present some modeling data to try to bridge these physiological metrics of drought and false spring vulnerability with what we know about maples already live, where maples already live, to understand how to use our understanding of how past climate change has shaped their distribution and forecast to some extent their likely response to future climate change. So I'll dive right into thinking about drought. And I'll just take a quick drink of water. So maples are complicated, large plants, and just like there's no one metric you can take of a human body to understand its well-being, for instance, blood pressure only tells you so much. There are many ways to quantify drought vulnerability within the world of maple water relations. I'm going to focus on this single one, Turgor loss point, or TLP, which can tell us quantitatively the point at which a leaf wilts. And that, interestingly, is often diagnostic how vulnerable its donor plant or mother plant is to drying if we were to deprive the entire plant of water. So to calculate Turgor loss point, we go through this complicated series of steps with a tool called a pressure bomb, often used in woody plant agronomy. There's also an indirect measurement that my lab is now using that I'm happy to talk about more. But basically what we are doing is cutting leaves off of plants and letting them dry out on the bench top of a lab bench indoors, and periodically measuring their water potential. And water potential is measured in units of pressure or tension. They're just the positive and negative version of the same thing. And plant physiologists by custom measure pressure or tension in megapascals. And a megapascal is about 150 PSI or 10 bars. Bike tires can be inflated to 0.75 megapascals, and car tires explode at 1.37 megapascals of pressure, so positive pressure. We are often measuring negative tension or suction because plant tissues are always under tension while they are alive, as water is pulled up from the soil through the plant into the atmosphere. So I'm, again, happy to chat more about this with anyone who's interested, but the takeaway from this is that plants with a high or less negative Turgor loss point wilt at relatively wet conditions and therefore are more vulnerable to drought. Plants with a low or a more negative Turgor loss point have to lose more water, relatively speaking, before they wilt and are more resilient to dry conditions. So here are some pictures of collecting and rehydrating leaves. This is a picture of a pressure bomb with a microscope head over it. So we just measure these leaves over and over on this bomb. And here's one of my former students, Buster, doing these measurements in the lab. We've now done thousands and thousands and thousands of these measurements over 3 years on a number of species of maples, as well as magnolias and hollies. So time to show you the data. I'm showing you one tiny little plot that I'm then going to multiply across many species, but I want to show it to you this way just to orient you to how to read it. So I'll just tell you one critical finding, which is that Turgor loss point, which it has been treated in the past as though it's a rather stationary measurement of a particular species or individual of the species, it turns out varies quite a bit over the course of the growing season. So I'm going to show you a number of plots in which the horizontal axis or x-axis is day of year, starting in about June and ending in August or September. So covering the growing season in Boston or Philadelphia, where I've done this research. And the y-axis is this metric we've just talked about, Turgor loss point. So remember, high numbers are wimpy trees that wilt easily and are not resilient to drought, and low numbers are tougher trees. Okay, so across the growing season from roughly early June to early September, I saw a consistent decline in Turgor loss point across almost every species we've measured, averaging at about 0.2 megapascals per month. So a really substantial change. And given this change, there was still a fair amount of diversity among species and among individuals within species. So I'm showing you first a data set collected at the Arnold Arboretum in 2019 and 2020. Green plots are East Asian maples, blue are North American, and red are Eurasian. And there are, each box is headed by the species name of the particular species. Maybe you'll see your favorite one in there. And within a region, so within a color, boxes are organized from most vulnerable to most resilient maple to drought. And I've drawn lines showing the tendency of maples to become more drought tolerant with time. What was puzzling and frustrating is that Acer triflorum, Acer saccharum, and Acer platanoides did not show this trend. And I thought it might be because for some of them we lost data points and maybe we were just capturing too much noise and not able to see a signal there. So with my students this last summer, I did a much more intensive measurement for each of these 3 species and did indeed find a pronounced drop in Turgor loss point for Acer triflorum and Acer saccharum. Acer platanoides, the Norway maple, is an interesting case. So it does show a very small decline in Turgor loss point over time, but it's quite weak, and in many individuals, it's absent. And this is curious to me because Acer platanoides, as many of you know, is considered a fairly invasive maple species from Eurasia. And in at least where I live right now, it tends to displace ecologically sugar maple, a species that we desire more for a variety of reasons. So the fact that it shows a different pattern relative to all of these other species, the only one of which is sometimes thought of as invasive as Acer tataricum, it was remarkable to me. But I want to also interpret what this decline in Turgor loss point over time means. So this is a signal of acclimation to dry conditions over time. As those of us who live in temperate, cool climates know, it is typical to have a pretty wet spring and early summer, followed by a progressively drying growing season, leading into a relatively dry fall and then more rain as plants are losing their leaves. And so this is my interpretation for this pattern, that maples with steeper slopes in these lines showing the relationship between time and Turgor loss point are those that are better able to adjust rapidly to conditions, and those with flatter lines are more static. Additionally, the further down in a plot that one line is located indicates that that species is relatively drought tolerant. Another interesting pattern to me was a continental pattern. So East Asian and North American maples, those on the top two rows plus one, varied among species in their drought tolerance, their Turgor loss point, but generally showed a fairly low level of drought tolerance and a high degree of diversity. Whereas the 4 Eurasian maples are much more resistant to drought, showing a really, when we consider temperate trees more broadly, a really high level of drought tolerance, especially in sycamore maple, field maple, and tatter maple. In particular, when we compare them to some of our native understory species like mountain maple or the striped bark maple, or to Acer caudatifolium or Davidii, these East Asian species grown horticulturally in the States. And this is a pattern that we'll see recapitulated elsewhere. So with this finding that many maple species that we're interested in ecologically or horticulturally are relatively vulnerable to drought, but that Eurasian species are less so, I'm going to turn briefly to vulnerability to these false spring events. And how do we measure this tendency to be in danger when one leaves out before winter has truly ended? So I'm going to present this technique it's often called differential thermal analysis or thermal analysis. So what harms plants principally when they experience a false spring is the freezing of water within their cells, of intercellular water. And this produces a thermal spike. Freezing water is an exothermic process. It paradoxically releases a little bit of heat when it happens. And this indicates that the underlying tissue in a plant has died. And these are close-ups from this Mills et al. Paper of grape buds. So if you chopped a bud off of the grape vine and took a close-up image of it, this shows a living and a freezing killed bud. So my colleague, Al Kowaleski, who's now at the University of Wisconsin-Madison, and I measured the temperature at which this freezing event occurred for 36 maple species in the Arnold Arboretum's collections. And we did so using this thermal analysis setup where we cut buds off of maple trees at different times of year and then progressively froze them in a deep freezer and measured the temperature at which they released that spike of energy, showing where they froze. And this is what data from this kind of analysis looks like. On the x-axis, we have declining temperature from freezing down to minus 40 degrees Celsius is generally thought to be the limit of how low many plants can keep water from freezing in their cells. And the point at which there's this sequence of spikes in the signature of the data shows us where water froze inside the buds placed inside of our device. And so for this particular toy example, it's minus 22 degrees Celsius, quite cold. So once again, I'll jump into our data and I'll show you data for a subset of 12 species for which we took measurements in February and March. Unfortunately, this experiment was truncated by COVID when our lab facilities closed. And we tracked this spike of energy indicating freezing for stem tissue, small twig tissue, and for buds. Buds are widely thought of as being the choke point for freezing damage because they are required, of course, for plants to produce leaves and flowers in the spring. And they're generally thought of as less resistant to freezing than stems, which agrees with what we found. So again, you'll see 2 points, one in February, one in March, and you'll see pink points for bud tissue and blue points for stem tissue. And this y-axis is going to be LTE, or low temperature exotherm, the point at which energy is released, indicating freezing. So this is a lot to look at, but the general patterns to notice are that indeed buds freeze at warmer temperatures than stems. Stems were almost always, although not exclusively, at the sort of, or close to the sort of maximum freezing threshold for plant tissue, whereas buds were much more vulnerable. And what we found was a pattern of loss in cold hardiness as we moved. Oh, these are from January to March. I misspoke before, not February. From January, the sort of deepest part of the winter when plants are maximally cold hardy, to a period when many of these maples are beginning to break bud and put out leaves and flowers. In particular, Acer caudata-folium, a Taiwanese maple, which was particularly vulnerable to drought, is also particularly vulnerable to freezing. Whereas some species, such as good old sugar maple, remarkably to me, retain cold hardiness at an extremely low temperature of -30°C, even in March. One species to note is Acer rubrum, which, as many of us know, flowers before it leaves out, and flowers in Boston in around February or March. And we can see its signature of very rapid loss of cold hardiness between January and March, associated with its bud break. So this may make it seem that maples are quite vulnerable to being frozen later in the season in March, when a fall spring might occur. But I want to kind of do a reality check, which is what I'm always doing to myself when I look at these data. I've shown in a light blue line across all of these plots here, the minimum temperature that we reached in March of 2020, and then the minimum March temperature in the longest running series of reliable records for Boston, which was 1872 is the coldest year on record for March, and which got down to about -22°. So as you can see, virtually all of the native maples and many of the exotic species planted at the Arnold Arboretum are still able to tolerate temperatures far below those that are either likely or possible to occur, especially when we consider the trend towards warmer and warmer winters and springs in this particular location. And so for Al, my colleague, and I, this translated to a low risk of fall spring damage for maples, especially in their native range. Risk for fall spring damage may be more of a consideration when growing exotic species in climates that are far colder, and particularly that have far more variable winter and spring climates than their native distribution, like Acer caudata-folium, which is from a fairly stable, almost subtropical climate, and when grown in Boston, is frequently knocked back due to freezing damage. So finally, I just want to share a little bit of this modeling work that I did when I wasn't allowed to be in the lab during our COVID, the beginning of our COVID times, and which begins to bring together some of our thinking about the evolution of the maple genus and its physiology. So I'm going to share results from a process called niche modeling that attempts to construct or estimate which factors of the climate are most important in structuring the native distribution of a particular maple species. So in this work, which I published this year in the journal Forests, I incorporated the genome-wide phylogeny from Arecis berezain, publicly available distribution data on maple species from the Global Biodiversity Information Facility, and then plugged them into this routine called MaxEnt, or maximum entropy species distribution modeling, that tells us where maples are most likely to be able to survive based on where we know they occur and don't occur. So I'm using this map of Acer circinatum, the vine maple, as an example. Black dots in this map indicate occurrences within its range. And the coloring of the background of the map indicates the likelihood that Acer circinatum could survive there based on where it occurs in its native range. Green and yellow and orange areas are more suitable, and red and white areas are less suitable. So I'll just skip over a lot of the nitty-gritty and share some take-home points. So there is very limited climatic niche overlap for the maples. Maples generally lived in different environments than one another. Even those that co-occur spatially seem to specialize in and occupy different microclimatic zones. There was a low level of phylogenetic signal or sort of evolutionary similarity among the niches occupied by these maples. So it was thought, for instance, that disjunct pairs of maples, those that have a pair that has one representative in North America and one in East Asia, would occupy similar habitats. That does not appear to be the case. What was interesting, though, was a pattern that I remembered from some of my physiological work. So here I've just summarized for the 3 maple continental floras, East Asia, North America, and Eurasia, the optimal temperature and precipitation regimes for all of the species for which I was able to get data. And what I found is, interestingly, across these 3 continents in which maple floras have been isolated for tens of millions of years, maples live in a fairly wide and uniform set of temperature conditions. So maples can basically be found in northern hemispheric forests, in which the mean annual temperature doesn't go below 3.5 degrees, and in which it doesn't get hotter than 17.5 degrees. This is uniform across all 3 continents. The story is different for precipitation. So in East Asia and North America, we see a similar story. Maples can survive with about 0.5 meter of precipitation every year, and they have colonized environments with up to 2 meters of precipitation every year, or 1.5 meters of precipitation every year in East Asia and North America, respectively. But it's in Europe and West Asia, where there are about 11 maple species, that we see an interesting divergence from this pattern. In this case, maples living in warmer, wetter environments are absent. They have been lost from the European maple flora. And this calls to mind, for me, the pattern in which we saw that European maples were on average more drought tolerant. So bringing these different lines of evidence together, I want to craft a story in which we think about the extent to which Europe and West Asia were glaciated during the most recent glacial periods, and in which what might have been once a more speciose, a more diverse, and a less drought-tolerant maple flora was driven to extinction, leaving behind only those species that could, for instance, survive migration to parts of Iberia or North Africa or the Middle East, and then a return to Europe during periods in which glaciers had receded. So in this sense, we could say that the Eurasian maple flora is depauperate, and in particular has lost species that could not tolerate hot, dry conditions. So I don't want to wave my hands too much, but I like to think about the implications for these findings in terms of threats, opportunities, and uncertainties for the maple flora in a changing world. So I'll just focus on the Eastern North American maples, because those are the ones that are a big part of my life right now. So overall, I think we should be most concerned about our understory and riparian species, those that have very high Turgor loss points, suggesting a low tolerance of drought, and which will likely face population pressure as our climate grows, as our climate becomes increasingly characterized by longer and more intense periods of drought. The prognosis is a bit better for widely distributed red maple and silver maple, which had pretty low Turgor loss points, and whose wide distribution suggests that they may be able to persist at least and even migrate to areas with suitable habitats, probably moving further north. Sugar maple is perhaps the lineage for which I think there is the most hope in our changing climate. Sugar maples, sensu lato, or in a broad sense, have survived past climate change by migrating to Mesoamerica and sojourning there, and then returning to further north to North America poleward as the climate has warmed. And there is considerable genetic diversity and even incipient speciation in this group of sugar maples, suggesting that at least some members of this group will survive at least moderate future climate change. So this is my foray into thinking about maples, perhaps in a different way than many of you have here. And I do want to just share why I tell other ecologists who aren't, for instance, maple enthusiasts, that this work is important, which is that maples are, as we saw, a key part of many Northern Hemispheric ecosystems. But I would also argue that they're in some ways a model lineage for thinking about the effects of global change and of climate change on our ecosystems. To some extent, what we find to be true for maples is likely to be generalizable to Quercus, Pinus, Betula, Salix populus, other genera that are interesting and important to us. So that's the pitch that I'll make, that these findings and our understanding and appreciation of maples go beyond just an interest in maples in and of themselves. So I want to acknowledge the very generous funding from the Arnold Arboretum, from my current institution, Swarthmore College, and the Scott Arboretum, and the many amazing colleagues, collaborators, and especially my students who have done this work along with me. And before I take questions, which I'm really happy to do, I want to encourage anyone who would like to get in touch with me at my work email address, which is printed below here, and which I'm sure Alan and others can pass on to you if you missed it. I will tell you if I'm a little bit pressed, but I do get back to emails as soon as I can, and I'm happy to chat more, to share versions of these results, and potentially to talk about ways for those of you who work with maples in a different context than I do, that we can collaborate, and that I can maybe help you find some ways of incorporating, of answering your questions through the incorporation of experiments or observations using the tools at my disposal. Okay, so I'll stop sharing my screen, and I guess I'll leave us recording for the question and answer session. I can see the chat again, so you're welcome to type questions or comments into the chat. You also can raise your hand using the Zoom raise hand function, and I will moderate and call on you. Or if you don't want to do any of that, you can just unmute yourself and go for it. So I really want to thank you all for taking some time to listen to and chat with me, especially for those of you for whom it's a bit late. It's really a pleasure, and I hope to hear from you further if any of this strikes your interest. So thank you so much. Yeah, Doug, go for it.
Jake, that was fantastic. Thank you. Have you visited the West Coast?
I did a master's in forestry at the University of Washington, and all of my in-laws are in Seattle. So I spent some time in Portland and Seattle, and a little bit in Vancouver.
Yeah, no, I just when you talk about this stuff and you talk about the eastern maples, it makes me think of, just how different the Western maples are.
Could you expand on that a little bit, please? I admit that I've just become totally biased from living in the East for so long.
Sure. Well, yeah, I mean, of course, drought tolerance. I mean, we go through, on the West Coast, particularly south of Vancouver, but including Vancouver, we go through a fairly serious drought in the summer, typically. And, things like big leaf maple, which, you would think with huge leaves and vigorous growth, long internodes, you would think that these things would be vulnerable as hell to drought, and they're not. They're tough, and Acer circinatum in the palmata section is probably the most drought tolerant of all, I think Acer glabrum it's an interesting case, because Acer glabrum kind of spans the Great Basin, and so the Californian variety glabrum are very drought tolerant, and the ones on the other side, on the eastern side of the Great Basin, are slightly less drought tolerant. And so when I see what you're doing, I desperately want you to do the same thing using Western North American maples. So let's see a much bigger data set.
I would love that. I mean, it's very, so I've been limited by my access to the plants. We have, when I left, we had 5 Acer macrophyllum at the Arnold Arboretum, and they get knocked back every year by freezing.
Yeah.
But, you know, if you, so I was just looking again at the slide, and, you know, the data I have for Acer macrophyllum really agree with your observation. Though they start off very intolerant of drought, they have a relatively steep slope of acclimation,
Yeah.
suggesting that they are able to respond to West Coast droughts. I would love to do this work. It just involves having access to trees you can clip bits off of, and you know, in some lab space. So it's very doable.
Yeah. Sure, I'll send you whatever you want.
Okay, that sounds good. There's some, you know, you have to keep them wet. So
Yeah.
we can chat more about that. I would really like to do that.
Okay, all right.
Yeah, Alan, go for it.
I'm muted.
You're muted.
Everybody's muted.
I'd like to ask Yoma Vargas about the Mexican maples and what her findings are as far as that goes, because definitely they're going to see some changes Central America and down in there. Are you there, Yoma?
Not muted, muted.
No. Oh, can I unmute her? Okay, there.
Yes, I am here.
Hello.
Hello.
Sorry, I don't have the camera, but I was actually in the field getting some data for Acer vinsayeda here in Western Mexico. But could you please repeat the question?
I guess I wanted to know how your findings with drought tolerance or spring early development follow through with what you have experienced in Mexico, because I know Central America in that area is going to receive a definite climate change action. Do you have any kind of idea of what the future looks like for the Mexican maples?
Well, actually, I have been tracking the seedling establishment right here in the forest, and the species and the trees only grows along a creek, small, very small river, and we have a lot of forest fires and some deforestation around. So the amount of water in the creek have decreased considerably. And actually, I was here trying to measure the abundance of the recently established or germinating the seedlings, and it is obviously a decrease. They are not tolerant to drought. Actually, it is a relationship with the amount of water that there is in the creeks and the percentage of germination here. So, but this is the case with the vinsayedi, which is in Western Mexico. And the one in the Eastern Mexico, which is Escuchay, it seems that it is a little bit more tolerant to drought. But anyway, it is related and only present along creeks and very protected ravines. And also there is a notorious relationship with the temperatures. Obviously, we don't have the minus 0 degrees temperatures here, but it seems that around 3 degrees Celsius is the most common temperature that facilitates the germination here. But currently I am doing some measures about that. So this is preliminary data, but I have been just continue measuring this, and I hope next year during the symposium I will present the data. I hope you can listen to me because my computer here is not working very good. Sorry.
Yalma, thank you for sharing. It's so great to hear that from you, and thank you for your amazing work.
Thanks.
I'm very humbled that you're here.
And actually, I want to thank the Maple Society because it provides funding for some of the devices that I am using now for the field for the sampling.
At this point, I'd like to point out that there will be a international symposium in October of next year in North Carolina. If you see anything that might be interesting, we'd certainly like you all to attend.
Robin, I see your comment in the chat. To be honest, I don't know exactly what to say. I'm not sure. So if you feel like saying more about how leaf scorch could be an adaptation to drought, that would be great. I do. So there is an observation, not necessarily for maples, but for many trees, that dry summers, dry, warm summers do tend to hasten leaf loss. And this, I think, is, maybe that's what you're referring to. That definitely seems like it's an adaptation. Well, it seems like it probably is a way of preventing hydraulic failure by getting rid of the leaves, which are, as Doug pointed out, the weak point in the plant when it comes to water loss. Is that satisfying or relevant?
Yes, I'm sorry, I just lost my Zoom connection. Can you still hear me?
Yes.
Oh, okay. I just lost my picture, I guess. I guess I would ask that, Doug, did you see the comment I posted?
Yes.
Is that your experience as well?
Well, I mean, it is, but I sort of imagine if you see along, driving down the highway and you see, 1,000 big leaf maples, and if those were Acer rubrum, they'd be fried, a month and a half before the big leaf maples. So, I look at it, yes, there's a certain point at which the maples can't tolerate the drought anymore. But, we can have 8 weeks of basically no rain, and big leaf maples still have leaves. I'd say that's pretty good. Midsummer.
Yeah, that was certainly true of this year.
Yeah, well, the heat dome certainly made mincemeat out of a lot of plants, but big leaf maples, despite even the heat dome, big leaf maples, still kept their leaves. I'd say that was pretty impressive.
So I'll add to the discussion that there's also this other dimension of drought tolerance that I have not gotten into in this talk, which is the observation that even if 2 species have the same turgor loss point, or sort of like drop dead point relative to drought, there is variation in the capacity to which different species can keep their bodies from reaching that point. So there's a sort of categorization of trees as plants as being drought tolerators versus avoiders versus escapists. So Aesculus californica might be an escapist that just loses its leaves. Acer macrophyllum might be an avoider that shuts down its leaves but keeps them to avoid dropping to perilous levels. Rubrum, in my experience, is to some extent a tolerator that allows itself to dry out, but plays a kind of risky game and assumes that rain will fall again. So there's this like other dimension that's orthogonal to everything I just presented, and that maybe can help explain why some species can accumulate damage or experience really dry conditions, but persist.
Jake, if I may, I have a question. In your data, I noticed you had Griseum as one of the species, which has a fairly narrow genetic base, whereas many of the other species you had have quite broad genetic. So is your data consistent with the idea that a fairly narrow genetic strain or species is similarly, I don't know what the word is, but adapted to drought conditions?
Well, that's a very perceptive question and one that I can't answer beyond a hunch. So, the data that you're seeing come from opportunistically sampled trees at either the Arnold Arboretum or the Scott Arboretum. So at the Arnold Arboretum, that includes two Acer Griseum that I believe are the source for a lot of the trees that have historically been in the nursery trade, plus two more recently collected ones that we might expect to be genetically distinct. The red maples, among others, span a much wider sample from a much wider pool, but it's the same number of plants, and there's no intentional sampling across the genetic diversity of each species. Yeah, I was actually quite surprised that the Acer Griseum that I've measured seemed very able to acclimate to drought. They did not show what you might expect to be the default, which is like a really maladapted kind of dysfunctional response to drought for a species that's gone through a bottleneck. That's all total speculation. I don't know anything about the genetics of Acer Griseum, and I don't know if anyone does in a way that's relevant to this question. So it's very tempting to say, wow, the Acer Griseum that have survived to this point and that were collected and propagated outside of East Asia are the ones that managed to stick it out. Yes, and so also, as Doug is pointing out, it does match the trait profile we would associate with being very resistant to drying. Those leaves are extremely leathery. They're pubescent, and so one hypothesis I was interested in at the start of this work was whether the compound leaf maples would be more resistant than the simple leaf maples, because leaf dissection and even toothing is thought to be an adaptation to avoid water loss. Acer negundo kind of blows that out of the water, but I'm interested to sample more compound leaf maples.
I was just going to type, but I don't type fast enough. Actually, compound leaves the adaptation is to radiative heat loss rather than drought tolerance. So the more dissected the leaf, the better radiator it is. So, but I don't think it has anything to do with actually drought tolerance. But if you're lining the stomata on the back of the leaf with wax or hairs, then that's a much better indication of drought tolerance. if you look at something like Acer henryi or more to the point, Acer sicifolium, those are very, very, dissected leaves or negundo, and they're primarily plants that require moist conditions. And, compared with Grisium, they're very, very different.
I wonder to what extent cuticle thickness plays a part.
Yeah, I mean, so it should be, it shouldn't be on a mechanistic basis related to this Turgor loss point measurement, but they are very likely to be highly correlated for this reason that we're discussing. They're both different but compatible ways of minimizing water stress. It may be of interest to note that Turgor loss point is probably that whether a plant has a high or a low Turgor loss point is probably a function to some extent of how rich in solutes or salts its cells are. So those that can, and that's probably how acclimation over the growing season occurs. The cells of these plants get loaded with salts with various dissolved compounds, and that makes them more resistant to giving up water.
I don't know if anybody noticed, but Alan sneakily is helping himself to turgidity
That's right.
monuments.
Keep my Turgor up. So
Yeah, that's right. I hope that's got a nice whiskey in it, Doug. Good man.
Well, again, I really encourage anyone who would like to get in touch with me by email, and I'm happy to share any of these results that have been published in the form of our papers, or to share some preliminary results as I very glacially process them and write them up. And again, I am, as I mentioned at the beginning, for those who are here, I'm in the process of trying to find a permanent academic home. But once I do, one of my priorities is finding ways to integrate this research with the interests and needs of producers and growers and public garden folks. So I'm really eager to hear ideas for collaborations or ways I can help out. Right now, I'm a little bit of a transient, but hopefully that will change soon. So I just want to thank you all again so much. And yeah, unless there are any other burning questions, maybe I can go to bed, as can other folks on the East Coast.
all right. Thank you very much, Jake.
You're welcome. It's really a pleasure. Thank you all.